Yes, yes, good morning. I'm Dan Davidson, and I will be moderating the webinar today. I want to welcome you to this morning's webinar on the Soil Advisor sponsored webinar titled Soil Biology Builds Resilience and Enhances Soil Quality, and it's brought to you by the Illinois Soybean Checkoff. Thank you for coming online today. Just a couple of details, housekeeping details for the day. If you included your CCA number when you registered for this webinar and you stay with us for the entire presentation, your number will be automatically submitted for one CEU in soil water management after the webinar is over. If you are listening to a recording of this webinar, you will need to go to the Certified Crop Advisor website, log in your account, and apply for a self-study credit. Again, one CEU in soil water management. And this webinar will be recorded and will be posted in the Soil Advisor's YouTube channel in just a matter of a couple days. You can ask questions during the webinar by submitting them using the chat feature in the dashboard on the right of your screen. We will, gather these questions together at the end of the webinar. We will pose those to our speaker, and she will answer them. So I encourage you to write some questions related to soil regeneration, soil health, as we go through the webinar. Without further delay, I'd like to introduce you to today's presenter, Dr. Kris Nichols. She's been a leader in the movement to regenerate soils for healthy crops, food, people, and planets. She is the founder and principal scientist of Kris, which is knowledge for regeneration and innovation in soils, and which is a systems education and consultation business. She's also a consulting research director for several companies involved in soil regeneration and soil health. She received a Bachelor of Science degree in plant pathology and genetics, cell biology from the University of Minnesota, a master's from West Virginia University, and a PhD in soil science from the University of Maryland. Kris has done a lot of work in soil health, pretty well known, particularly with some of her work with MYCORRHIZAL and the soil protein gomalin. That's one of the byproducts of MYCORRHIZAL. And we look forward to hearing her speak on her view and vision of soil health and all the microbes that make it possible. Kris, I'll let you take it from here and go through your presentation. When you're done, then we'll do a little Q&A.
Thank you so much, Dan. Thanks to Dan and Jill. And I am very happy to be able to speak with you all this morning to talk about one of my favorite subjects, which is soil biology and how that's related to soil health and building resilience in your system. This summer has definitely been a challenging time for most people, but I think that it is also a thing that we need to be looking at to get a better understanding of how we need to be approaching our future. So what I want to focus on with soil health is the idea of soil function and what's happening with soil function, that we're able to build resilience and resistance and also improve the nutritive quality and the overall profitability of your system when it is that we're looking for functions that are going to come from the biological components in our soils. When we think about the biological components in our soils, one of the things that we need to do is take a systems approach. This isn't looking at one tool, one mechanical or chemical tool to be able to resolve a particular issue, but it's looking at what's happening within the entire system. So what we want to do is we want to be able to tap into photosynthetic activity. Photosynthesis is the most efficient form of solar energy conversion to chemical energy and holding that energy in the bonds between carbon atoms and other atoms. So it's really key for us to be able to utilize photosynthesis as the main way to be able to tap into what's happening within our system. Now, what we want to do is we want to focus in on what I see as basically the the root of the problem. I think that there's really one problem in agriculture, and that problem is a lack of carbon. Everything else that we find as challenges are actually issues. If it's too hot, too cold, too wet, too dry, my soil's too heavy, my soil's too sandy, it's too low in fertility, all of those things are issues that can be resolved if we focus in on the problem. And the problem is that we don't have enough carbon in our soil. And so when we look at this diagram of the soil food web, I like to put this up because a lot of people will focus on what's going on sort of in the middle of the screen, looking at the food web and the interaction of all of the organisms in that food web. But what we really need to focus on is the fact that without the first trophic level, without photosynthesis, we actually don't have a food web. You need to have photosynthetic activity in order to have soil biology. In fact, you need to have photosynthetic activity in order to have soil itself. So we need to be focusing again on increasing the amount of photosynthetic activity that we can have in our system as a foundation for what's going to be able to occur. So again, what we're looking at is the fact that our soils are really deficient in carbon. That is our problem. The carbon problem is what it is that we need to focus in on and solve. And you see here two different soils. Some of you have probably heard of David Brandt, and hopefully some of you have been down to David Brandt's farm and been to his field day. What you can see here is a picture of David Brandt's soil and a picture of the soil that is, according to NRCS, the soil that David Brandt should have. This is the soil that's right on the other side of the fence line. You can see that what David has done over the years that he has been working on his farm is he's been able to regenerate and recarbonize that soil. He's been able to put carbon back into that soil environment and build up the resiliency in that soil environment and build up resilience in soil health, so that what you have is resilience and resistance against all different sorts of issues that might arise. Again, whether it's climatic issues such as precipitation or temperature, or it's fertility issues, or even pest and disease issues. You're able to manage those issues much better when it is that you have addressed the carbon problem. So what we want to do is we want to have a new revolution in agriculture. Instead of having a new green revolution, we actually need to have a brown revolution or a soil revolution, where we're going to be focusing on eco-functional intensification. What this means is that we're not going to necessarily repeat everything that nature does. What we're going to do is we're actually going to utilize those natural tools to be able to try and address some issues that it is that we might start to see. We're going to utilize natural tools to give us ideas on how we can move things forward. But we're not going to totally repeat what everything that nature does. We're going to look at all of your farmscape and determine what might be some of the better eco-functions for those areas on your farmscape. So doing things like being able to look at an area of land where maybe you don't get the highest level of production, but it's an area that is frequently wet. Can you take that out of production and put that into some sort of perennial production that's going to enhance pollinator populations or be utilized for grazing, providing yourself with some other options that you can put in to that landscape, looking at its eco-function. But at the same time, we're going to intensify what we're doing. What we want to do, again, in looking at what nature has done, is natural organisms, you, me, mycorrhizal fungi, plants, cattle, birds, bats, bees, all organisms are pre-programmed to be lazy. It's true. We're all programmed to be lazy. What that means from an ecosystem level or from a programming level is we're pre-programmed to conserve resources. It's efficiency of use of our resources. So what we want to do is we want to not have a system that is constantly going to be lazy. A natural system responds to stress events and increases efficiency. But most times it's going to be functioning at a low, moderate level. It's going to be lazy. And then when you have a stress event, such as a fire or a swarm of insects or major grazing event, any of those types of stressors, then a natural system will kick up its intensity, and every organism within the system will work at a much higher level until the system gets back to an equilibrium state and then can go back to being lazy. What we want to do in our agroecosystems is we want to keep them functioning at that level of peak efficiency. And that's going to allow us to be able to get more out of the system. But what we want is not to have to do more work in this system, but do less. We're going to rely on the entire system, on every trophic level that's involved, on the plants and on the animals, whether it is animals that you're bringing onto the landscape or it's animals that are coming onto the landscape naturally. Things like various types of insects, pollinators, bats, birds, all of these organisms that are going to come in and be a part of that landscape. We also, in that process, are going to be creating multiple enterprises that you can utilize to be able to enhance your income. But what we need to do with those multiple enterprises, recognize that there is a cost to everything that we're doing. This isn't magic. Soil biology isn't a magic cure-all. It isn't a thing that you can add and poof, everything's going to go great. It is looking at how we're going to redistribute the risk and redistribute the cost among all of the different plants that we may be growing in that system. And what this is going to do is not only can this help enhance our productivity, but it can also enhance the nutritive quality of the products that we're growing. Many of the nutrients that we're looking for from food, not just the mineral nutrients, but things like antioxidants and polyphenolics. These things that we're now having to take supplements for, those molecules are actually stress biomolecules. The plants create them in response to stress. So if we're not putting stress in the system, we're not going to get those biomolecules. So what I've done here is I've taken the principles that have come across for principles for soil health. There's about 5 to 6 principles that people talk about for soil health. And I put them in this pyramid design. And the reason that I put this in this pyramid design is for 2 critical points. One is when you think about the pyramids have actually stood for thousands of years. And they've stood for thousands of years not because the capstone or the layer at the top is any more important than the layers below. Pyramids that were not completed, those are the pyramids that fall sooner than the pyramids that were completed. So if you look at archaeological records, you'll see that those pyramids that were completed that have every layer in the pyramid playing a role. So when we think about this pyramid, this isn't about hierarchy. Reduction or no-till systems aren't any more important than managing livestock or increasing diversity or reducing your inputs or adding armor. And again, it's important to be able to look at how every layer is integrated and interlocked. That's what's helped these pyramids to be able to stand for the thousands of years that they've stood. And what I want out of this, my other point with this, is that I don't want you to be able to just celebrate a century farm, but I want you to celebrate a millennial farm. I want you to be able to be on the land for thousands of years. I come from a farming background in Minnesota. My dad's owned the farm that he currently is still farming for my whole entire life. He purchased it the year I was born. And it's important to have, coming from a farming background, recognizing that linkage that you have with the land. And I want you to be able to stay on your land, again, for thousands of years, not just a few years here and there. Currently in the United States, we lose about 1.6 billion, with a B, metric tons of topsoil every year. What this means is if you were to load all of that topsoil in boxcars, the length of the train would actually stretch around the equator up to 7 times. So we're losing our land. We're losing our soil. We need to be putting these principles in place in order to be able to regenerate our soil. So what do some of these principles mean, and how are they linked with soil biology? Well, another way to take a look at this is actually to take a look at this from the point of view of treating the soil like you want to treat yourself. Think about the things that you need to do to be a healthy organism. The soil itself is kind of like an organism. It's all of these organisms interrelate and interlock and form these consortia, and so it acts like an organism itself. So one of the things that we're supposed to do if we're going to be healthy is we're supposed to eat small meals throughout the day. We're supposed to be grazers. Well, if we're supposed to be grazers and eat small meals throughout the day, it means we have to have access to food throughout the day. Access to food is through carbon, access to carbon. And so in our agroecosystems, we need to have plants growing for as long as possible. So in Illinois, you need to have plants growing for at least 260, I would say closer to 280 or 290 days out of the year. That's how often you need to have something growing in your crop field or in your pastures. And in your pastures, most often, you are going to have plants that are going to be growing for a longer period of time. But what we want is we want to be able to have plants growing as long as possible to feed the soil. So it isn't just about growing a crop plant, but it's also about being able to grow something that's going to feed the soil. And if you think about it, the sun shines every day. If we're taking and activating photosynthetic activity, the important key with this is to be able to have something growing as much as possible. Now, not every plant is going to be able to grow at every temperature. We're going to have different plants that are adapted to different temperature regimes. But plants are not like us. They don't have a nervous system. So they don't sense temperature in the same way that we do. They actually have a fairly wide range of temperatures in which they can grow. So what we need to be looking for are plants that are going to be able to grow at different times of the year and be able to integrate those into our systems so that we're able to maximize that. If you're only growing a plant for 260 days, you're taking 105 days off. That's 105 days of vacation every year that the soil is not getting fed. If we want it to be healthy and we want to be feeding it on a consistent basis, small meals, giving it carbon all the time, we need to make sure that we're maximizing the amount of time that we're putting carbon below ground. Now, the other thing with that is naturally what comes in is diversity. So when we looked at that pyramid, the bottom layer on the pyramid was about keeping it green and growing. That's keeping the plants there for as long as possible. Diversity naturally is interlocked or integrated into that because in order to be able to do that, you need to have diverse plants. Yeah, you're not going to have corn or soybeans that's going to be growing 260 days. What we're looking for is, again, adding other plants into the system as cover crops, as companion crops, as polycropping, anything that you can do to maximize the amount of plants that you have growing in the system. Again, intensifying your system. The other thing that we're supposed to do to be healthy is to exercise. We don't want to over-exercise the system, so we want to add a little bit of stress. If you want to be a gold medal winning Olympic athlete, if you want your soils to perform like gold medal winning Olympic athletes, you need to exercise your soils. You need to put the system under a certain amount of stress. And stress for the system involves things like grazing, whether the grazing is happening from an animal that you're introducing, or grazing is happening with insects, or grazing is happening with other animals that are coming in. That little bit of stressor that's happening to the plants are connected below ground, and oftentimes if one plant is stressed, signals can get passed between the plants through the soil biology, especially the mycorrhizal fungi, to tell them that that stress is coming. And so then they build those stress biomolecules, those antioxidants and polyphenolics. If we want to get the healthy things that we need from our food, and those stress biomolecules are important to the plants because they protect the plant. They protect and improve the integrity of the plant cell wall. Just like for us in eating them, it improves the integrity of our cell membrane. So it helps to protect us. So we want to have a little bit of stress in our system, maybe a little bit of competition from a companion crop. You want to be able to have that stress. You want it to happen at the right time. So you need to think about when you're helping to introduce that stress, the intensity and the scale and the frequency of that stress. So you don't want to overdo it. Again, an athlete will exercise, but if they overstress themselves, they're not going to be Olympic gold medal winning Olympic athletes. They're not going to be able to continue to perform. The other thing you want to do is you want to protect your body. You want to protect the soil from injury, from radiation, and from temperature extremes. That, for the soil, is putting on the armor, keeping the residue that's on the surface of the soil, making sure that the soil is protected so that it doesn't get exposed to the solar radiation, to the heat intensity that comes from sunlight. You don't lose a lot of water to evaporation. You want to make sure that you're protecting the soil from erosive losses, from wind and water erosion. It's estimated that the intensity, the amount of energy that comes from a rainstorm on an acre of land is up to 20 tons of TNT. These are little explosive forces that are hitting the surface of our soil. What we want to do is we want to make sure that that soil is protected. You can protect it with crop residue. You can protect it with mulches, but you can also protect it with living plants. The energy that comes from the rain, solar radiation energy, all of those energy forces are going to hit the residue or the leaves of a growing plant before they hit the soil surface. And so some of that energy gets lost before it hits that soil surface. And that's going to help to protect the soil. So again, it's integrating the idea of treating the soil like you want to treat yourself, or the principles of regenerative agriculture. Both of them can be thought of as working together. So one of the important things too within the principles is the idea of reducing the amount of synthetic inputs. One of the reasons we want to do this is we want to improve our nutrient use efficiency. Again, this is about improving the efficiencies of the system. So what these graphs show, the top graph is an increase in global cereal yield since 1960. And so we've had an increase in our yields with breeding and genetic programs that we've had with our crops, which is a great thing. It's helped to support our population. But the bottom curve is actually a nitrogen use efficiency curve. What it is the amount of bushels that, the amount of nitrogen that it takes to produce a bushel of grain. And so what has happened is that curve has gone down. And what this means is that since 1960, it takes more nitrogen today to produce a bushel of grain than it took in 1960. And it's not just true for nitrogen, but it's also true for other nutrients that you have in the system. So we've always known that when it comes to synthetic nutrient use, the synthetic nutrient use of things like nitrogen fertilizer is somewhere around 30 to 50%, meaning that about 30 to 50% of the nitrogen fertilizer that you apply actually ends up in your crop plant. For phosphorus, it's about 30%, and in some cases, there's been data that's shown it's gone down to about 10%. This is because those nutrients aren't going to stay in a continuous available form for the plant. We apply them in a form that the plants can take up, but this is basically like going out and buying a goldfish, and you buy the goldfish, and the fish food is on sale. So you buy the fish food as well. And you basically go home and you're like, you know what, I can't feed this fish every day. So what I'm going to do is I'm just going to put the amount of food in the aquarium that the fish needs for the whole entire week. And I'll do that every week. I'll just dump that food in. Fish food's on sale. It was cheap. What we do is we do that in our agroecosystems as well. If you were to do that to the goldfish, you're going to come back just a couple of days later and the goldfish is going to be dead because the fish doesn't know how to manage the food that's there. The fish will overeat, and it may look good for a short period of time, but then it can't eat anymore. And it's going to start to starve because part of that food is going to get broken down and not going to be available to the fish anymore. So we need to look at how it is that we could time our nutrient addition so that we're doing it when the plant needs it most. The plants have their highest nutrient demand when they're going into reproduction. And so we want a time to get most of the nutrients into the soil when the plant is going through reproduction. But that's really difficult for us to be able to do. It's hard for us to get out there as far as equipment is concerned, and it's difficult for us to get out there during the reproductive phase because we've got other things that we're doing at that time. So it doesn't end up working out very well for us. But are there ways in which we can spread out some of the nutrient application? And more importantly, can we utilize the activities of the microorganisms? In 1960, before we were adding a lot of synthetic inputs, we were actually utilizing the activities of the microorganisms, which are able to free up and release the nutrients from the soil over time. So what we want to do with this is we want to actually tap into an interactive carbon economy. We want to have this idea of carbon, of food, being traded from the plants to fungi and bacteria, to organisms that are in the soil. And you have organisms like mycorrhizal fungi. Dan mentioned I've worked with mycorrhizal fungi all of my adult life, essentially. And these are organisms. Myco is the Greek word for fungus, and rhiz is the Greek word for root. And so they're basically fungus root fungi. And what they do, the fungi, their bodies are these fine threads that you'll see in the soil. And these fine threads are actually linking the soil environment to the plant roots because they'll grow inside the plant roots themselves. And this is really critically important because the nutrients are in the soil. Even if you do a really good job with fertilizer placement and you want it near the growing plant roots, you can't put it too close to the seed, but you want it close to where the roots are going to grow into it. So you're going to have the nutrients there. But this isn't like Star Trek where the plant can say to the nutrients that are out in the soil, I'm just going to beam you in to my plant. We can't beam the nutrients in. The nutrients have to move from the soil environment and they flow towards the plant roots. And the plants can help to trigger this by having around their rhizosphere, it is a more moist environment. They're giving off water and exudates in order to create a moist environment around the rhizosphere, and that's going to allow for the nutrients to move from the surrounding soil by diffusion. So they're able to move through the water and diffuse into an environment around the roots where they're in lower concentrations and they're in higher concentrations out in the soil. But this can be a fairly inefficient way of being able to move nutrients if the nutrients are spaced further away from you. So, you know, this is similar to, I look at this in a similar way to your farming in Illinois, and for a short period of time, there's food in Illinois, but all of a sudden, because of the fact that the people that live in Illinois, all of a sudden, there's going to be no more food left in Illinois. And so you have to get food now from Minneapolis. And how is it that you're going to get the food to come from Minneapolis to Illinois? One of the things that you could do is you could put an interstate from Illinois to Minneapolis to try and get that food to be shipped into Illinois, but that shipping program is only going to work to bring the food that is right around the interstate that comes to the interstate in Minneapolis. Wouldn't it be more efficient if you could actually have side roads that would come off of the interstate that would actually go out into all of the different neighborhoods in Minneapolis and knock on everybody's door and be able to pick up all of that food and bring it into Illinois? And so it's really important for us to be able to take that same idea in the soil. It may only be an inch or two away from the roots, but for the plants and for the plant roots, this again is like the food being in Minneapolis. So it's a further distance than what you think about it. So what we need is we need to have a way of being able to access. And to do that, you have these fine threads that come out of the roots, like all of the side rows, and are able to access more of the soil environment. You also have other organisms that are going to be able to fix nitrogen. You have the nitrogen fixers that are with legumes like soybeans, and you have free-living nitrogen fixers as well. So you've got these organisms that are actually going to be able to take nitrogen from the air and convert it into nitrogen that the plant can utilize. In addition, you have bacteria that are able to solubilize phosphorus. Phosphorus is normally not very available in our soils, but there's a lot of phosphorus in our soils. It's just not available. And that's the case for most of the nutrients. We live in an environment that has everything the plant needs to be able to grow, but just not in an available form. It's kind of like as if you were stranded on the ocean, surrounded by all the water that you wanted, but not a drop to drink. We need to be able to access more of that environment, be able to tap into this and utilize the organisms that are going to help to break down those nutrients. You're also going to have larger organisms like nematodes and protozoa that are going to eat bacteria and fungi. And when they do that, they're going to release waste. And in waste, like manure, in any waste, there is nitrogen and other nutrients. And so you're going to get the cycling of nutrients within the system that's going to happen. One of the key organisms that I mentioned are the mycorrhizal fungi. These mycorrhizal fungi can obtain up to 90% of the nitrogen and phosphorus that a plant needs because they're going to work with these phosphate solubilizing bacteria. And they're also going to have mixed cultures of different species of mycorrhizal fungi that are going to be adapted to your soil environment. And it's not just nitrogen and phosphorus that mycorrhizal fungi can be involved in. They can also help to obtain water. They can help to obtain micronutrients, copper and zinc and boron and molybdenum. all of these different elements that we find sometimes that are limiting in our system can actually be transported by the mycorrhizal fungi. They're fine threads that their bodies are made of, this hyphae that goes out into the soil, as you can see in this picture, and picks up those nutrients and acts like a pipeline, carrying them in to these structures inside their roots called arbuscules. And they're highly branched, just like trees, arbor. They're highly branched structures that are out there, that are out in the soil, but highly branched structures, the arbuscules, are also inside the roots, and that increases the surface area, the exposure of the fungal hyphae to the root cell membrane. And so you get a greater level of exchange. If I had just nutrient exchange that was occurring and I didn't have my fingers branched out, this would be all of the exposure that I would have to the membrane. But if I have my hand all branched out, I'm going to have a greater level of exposure to the membrane, and I'm able to more efficiently transfer nutrients and water. It's also found that, again, these mycorrhizal fungi, these organisms, can induce the formation of various types of antioxidants, these stress biomolecules. Now, in doing that, one of the things that they can do in triggering that in the roots is that can help to prevent various other types of pests and disease organisms from being able to enter the roots. If the roots are colonized by the mycorrhizal fungi first, they're less likely to be colonized by other fungi or less likely to be attacked by bacteria or insects. So it's a way to be able to help to build up the resilience and resistance against pests and diseases. Now, one of the other cool things about the mycorrhizal fungi is that they're able to help to transfer nutrients between two different plants. So what you have here is you have essentially a legume plant like soybeans that is also growing with a non-legume. If you had something like soybeans and wheat in the same field, what you can have is you can have a nutrient transfer of nitrogen from the soybean into the wheat plant through the mycorrhizal fungi. And the way that this works is we're going to have 5 organisms involved. We've got the 2 plants, the legume plant and the non-legume plant. We've got 2 bacteria, the nitrogen-fixing bacteria, and we've got a phosphate solubilizing bacteria, and then we've got the mycorrhizal fungi. And so what happens is the non-legume plant is looking for both nitrogen and phosphorus. And in looking for nitrogen and phosphorus, it essentially asks the mycorrhizal fungus to go out there and get nitrogen and phosphorus for it. The mycorrhizal fungus says, you know, I can do that. All I've got to do is I can go into this legume plant that seems to have a lot of excess nitrogen, and I can bring some of that nitrogen back for you. And I also seem to have these phosphate solubilizing bacteria that live on my hyphae, that live just outside my door, where they're going to process phosphate and release that, and I can pick up that phosphate and I can bring it to you. So the mycorrhizal fungus then goes over to the legume plant and then asks the legume plant for some of the nitrogen. The legume plant says, all right, I will give you some nitrogen, but you have to give me phosphorus in order to be able to reach my demands and also some of the other micronutrients that I need. And the mycorrhizal fungus says, I can do that because I associate with the phosphate solubilizing bacteria. So the legume plant then goes down to the rhizobium bacteria that are in its roots, and the legume plant says to the rhizobium bacteria, you need to do more nitrogen fixation because I have to have nitrogen for myself, but I also need to give nitrogen to the mycorrhizal fungus, so the mycorrhizal fungus can give some of that nitrogen to the non-legume plant. Now, the rhizobium bacteria says to the legume plant, well, I can do that, but I need more phosphorus in order to be able to do that. So it takes the cycle, in order to be able to get the energy to do nitrogen fixation, it takes this cycle called the Krebs cycle. And in the Krebs cycle, you have a molecule called ATP, adenosine triphosphate. And there are 3 phosphate groups that are bound to the adenosine molecule. And what happens is when one of the phosphate groups gets ripped off, it becomes ADP, or adenosine diphosphate. And that ripping off of that phosphate group releases electrons. And electrons then provide energy, or essentially fire the cell so that the rhizobium, so that it can fix nitrogen. Now you need to have 16 of these cycles, and you can't take the phosphate that got ripped off and put that back on and make ATP again. You have to actually have new phosphate come in and bind to the ADP, adenosine diphosphate, and make it ATP, adenosine triphosphate. So in this process, you have all of these things that are going on. So it's going to take a lot more phosphate in order for this to be able to occur, because you need to have 16 of these cycles for this to happen. So we need more phosphate. So the legume plant then goes back to the mycorrhizal fungus and it says, you have to get me more phosphate. And so the mycorrhizal fungus says, all right, you know, I can do that. You give me some carbon. I can give some of that carbon to phosphate solubilizing bacteria. They're going to make the enzymes in order to be able to release the phosphate. That phosphate can go through me into the non-legume plant, and it can also go through me into you, and then you can give some of that phosphate to the rhizobium, so the rhizobium can fix more nitrogen. The nitrogen can then go into you, and that nitrogen can then go from you into me, and then I can take some of that nitrogen and I can give that to the legume plant. And all of this is going to be happening simultaneously in the soil environment for these nutrients to be able to feed the different plants when they have those demands. When we can utilize this and we get the growth and production of the mycorrhizal fungi in the system, not only is it important for nutrient and water movement, but it's also important for water infiltration and water holding capacity. It's allowing us to be able to build resilience so that we can thrive at different times. So what you have here is a picture of Gabe Brown's ranch near Bismarck, North Dakota. And this is the next day after we had a rainfall event where we had 13.6 inches of rainfall in about a 24-hour period. How many of you can say that you could take in 13.6 inches of precipitation in 24 hours? This is because Gabe has improved his soil structure. He's improved the activity of his biology that has allowed him to take his infiltration rate from a ½ an inch per hour in 1991 to over 8 inches per hour in 2011. It's because he's improved the porosity. Now, how do we improve porosity? Well, we improve porosity by improving the formation of soil aggregates. Soil aggregates are these pellets in the soil, and they're created by forming around a frame that's created by the fungal hyphae. So you essentially have sand particles and silt particles and clay minerals, and you have organic matter and debris that starts to collect around the fungal hyphae around the frame. So it's like a frame in a house, and you start to get debris that's going to be collected between the frame. It starts to build up the soil aggregate. Now, one of the important things with the aggregate is you want to have molecules that are going to help to hold those particles in. So some sticky substances, polysaccharides produced by bacteria, and a molecule called glomalin that's produced by mycorrhizal fungi that can help to stick that in. In addition, glomalin has both a sticky side and a waxy side. And that waxy side is like putting weather sealant on the outside of the aggregate that you've created. And that's what that picture in green shows. Is it's basically a laboratory procedure where we stain the glomalin so that when we put it under blue light, it's going to glow green. And we can see it on the surface of the aggregate. And that's going to put that weather sealant or that coating on the aggregate. And so what you have normally is you have an aggregate in the soil, and when it's dry, there's in that gray space, it's filled with air. Now, when water comes in, the water rushes in through all of the openings at the surface of the aggregate, and it comes in and it moves at a faster rate than the air can move. Because of density, water is a liquid and air is a gas, so the water will move at a faster rate. It comes in very, very quickly, and it pushes those air molecules closer and closer together. And what you end up getting eventually is enough air pressure that the aggregate literally explodes. What we want to do is we want to take that aggregate and we want to put some weather sealant on some of the openings on the surface. So there are fewer openings, so the water comes in at a much slower rate, and now you can have space within the aggregate that is going to be water filled. and the aggregate is still going to stay together because the air has time to escape. The air can move through the weather sealant coating, but the water cannot. So the air has time to escape. We see this in our soils. So these are soils from a research study. The first two are sites that were side by side. The next one is about 3/4 of a mile away. What we had here is a conventionally tilled spring wheat fallow system, a no-till spring wheat, winter wheat sunflower system, and a moderately grazed pasture. And all that I did was I separated the aggregates out by passing the soil through a series of screens, and then I just added water. And that's what you see at the bottom. WSA is the value of the aggregate stability. But you can visually see the difference that happens. So in the first one, we had conventional till spring wheat fallow, meaning we had a crop growing once every other year, and it was spring wheat, and we did a lot of tillage. So we changed that. We decided we needed to have a crop growing every year, and we needed to have a little bit of diversity to our crop, and we also did no-till. So we applied a number of the principles to this process, and we improved the aggregate stability, but we still hadn't improved it as much as when we have a pasture system. And part of the pasture system is what can happen with grazing, but it also is the fact that we have plants that are growing for a longer period of time. So in a spring wheat, winter wheat, sunflower system, we didn't have enough plant growing, we didn't have enough photosynthesis. So we see similar increases in aggregate stability when it is that you have plants, if we incorporate cover crops or polycropping or companion cropping. So all of these can be beneficial to the system in order to be able to get things functioning at a higher level. So again, what we want to do is we want to get this brown revolution going. We need to figure out ways in which we can increase the amount of plant productivity that we have, so we can increase the diversity and increase the biological activity by reducing the synthetic inputs. If we utilize synthetic inputs, what it does is it outsources the job of the microbes. And if they don't have a job, they don't get paid, and their pay is food. So that's what happened from 1960 to 2000, is we outsourced their job. They didn't get food. They couldn't grow and reproduce. And so we've lost biological activity over time. We want to include livestock, whether that is an animal that we bring onto the landscape or helping to integrate animals that are coming onto the landscape. Things like birds and bats and bees and insects, and even other animals, natural animals that will come in and graze, and small mammals, all of these things. Now, the other thing is again, we want to protect the soil surface from the impact of solar radiation and raindrops, and we want to reduce the disturbance of the soil. We want to make sure that we're keeping that soil protected, we're keeping that structure intact so that water can get into our soil at a much quicker rate, and we're also able to hold water. When you have that increase in aggregation, the aggregates can't fit tightly up against each other. And so what that does is that creates open space around the aggregate. It's like putting a curvy road in the soil. And so when you have a curvy road in the soil, you can take more water following that curvy path. It also is important in that curvy path because it's harder for the sun, for evaporation, to pull that water out from the surface, or gravity to pull that water down. So we're going to increase our water holding capacity as well as our infiltration rate. One of the things that we need to be thinking about as we move into the future too is how we're going to improve the nutritive quality of what it is that we're growing. So when you look at your new consumer, at your new customer, this is who she is. This is where we're going in the future. And this person is willing to pay more for foods that she sees as being beneficial. And in order for those foods to be beneficial, they need to have higher levels of minerals. They need to have higher levels of antioxidants and polyphenolics. And for those to get into the system, they come in through these types of stresses. So what I want you to be thinking about is how everything within the system is interlocked. What it really boils down to is this, that everything is, that all life is interlocked, that all life is interrelated. We are all caught in an inescapable network of mutuality, tied into a single garment of destiny. Whatever affects one destiny affects all indirectly. We need to look at how the system can function together. And with that, I want to thank you and open it up for questions.
Kris, this is Dan. I got some questions here. I do want to comment. I thought that was a very interesting description. Somebody would ask me what the number one thing I learned here. I'm just going to describe what you described. You know, and most aggregates in the soil will absorb water. As the air is expelled, it will explode. But, you know, aggregates with this squamolin weather sealant, as it takes up the water, that is a little bit more flexible, and that aggregate will stay intact as the air escapes. I thought that was a great description. I never got that part, so thank you very much. Did I convey that correctly?
Yes. Yeah. Thank you.
Hey, so how did you measure aggregate stability in that one slide you had? And can you just kind of define, were you measuring macro or micro aggregate stability, and what is the difference?
Okay, that's a very good question. So what I've done here is I'm looking at what are called 1 to 2 millimeter aggregates. They're macro aggregates. And essentially what I did was I took the soil and I passed it through a series of screens. So a screen that had a 2 millimeter opening, and then the soil that passed through that screen, I would collect that soil and I would run it through a screen with a 1 millimeter opening, and what stayed on the screen is the 1 to 2 millimeter aggregate. So they're larger than 1 millimeter and smaller than 2 millimeter. Now, if you wanted to do this yourself, and you don't want to spend the money on buying sieves with the exact screens, you can actually go out and get screens that are pretty similar to 1 to 2 millimeter. A standard window screen is about a 2 millimeter opening, and if you get a very fine mesh screen, it's usually referred to as things like no-seam or very fine mesh. That is the no-seam, so it's things to keep out, you know, really small mosquitoes and gnats, or gnats and other insects like that. That is about 1 millimeter opening. So what I've done is you can sort of build these yourself. You can use PVC pipe or any type of tool like that and glue the screen to the bottom of that, and then pass the soil through the standard window screen, and then collect that soil, put it through the 1 millimeter, the no-seam, and collect that soil. And then if you take, so to get the measurement, it's by weight. So I take a weighed sample of the aggregate, and I'll put them back on the 1 millimeter screen, and then I dunk them in water, and what stays on the screen, the weight of the aggregates that stay on the screen, I'll measure that. Now, not everything is an aggregate. You could have sand grains or soil particles or other things like that. So what I'll do is then I will take what stayed on that 1 millimeter screen, and I will basically put it in soapy water, and that will destroy the aggregates. And so the aggregates will go through the screen, and then what stays on the screen will be sand grains or organic matter or other things like that, and I weigh that. And so if I have my initial weight, and I take then the weight of the aggregates that stayed on the screen after I dunked them, and subtract out the sand particles and debris, and then I divide that by the initial weight, that gives me the percentage of aggregates that are water stable.
Right. Okay. Okay. You know, moving on, we don't have a lot of time. The microbiome is a community of thriving organisms that are sort of adapted to survive together, right? Can we economically or practically shift that community? You know, I'm thinking of all these biologicals that come to market, right, that are make some claims. Can we even shift that community? Is that even feasible?
Well, there are a couple of issues in trying to shift the community. One is our basic ignorance of what the community should look like. Essentially, and this is a very true story, but the reality of it is this: when I started at the University of Minnesota, we thought we knew about 10% of the organisms that were in the soil. We could culture them, identify them, we knew what they did. By the time I finished my doctorate, about 12 years later at the University of Maryland, that number was down to 0.1%. So essentially, wow. I became 100 times stupider in 12 years of college. And so this is what we're learning, is that there are a whole lot more organisms in the soil than we ever thought. And that number keeps growing as to the number of organisms. And so if you do like microbiological testing, frequently they'll have a certain percentage of that microbiological community. They'll give you information about the different organisms that are there, but they'll have a certain percentage of what they analyze, which is unidentified. And oftentimes that percentage is somewhere 40 to 60%. It's unidentified. We don't know what it is. We also, when they work as a community, we don't understand all of their functions. So they change functional roles. So it's difficult to be able to shift the community, to know what community you should shift it into. And then when the functional roles change, how does that work? So I always encourage farmers to do things that are basically going to enhance more of the community that's there. And that's why I talk a lot about increasing the amount of plant cover that you have and following those principles.
I think that goes to a question another question just came in. I think you partly answered that. I'll just read the question off to you. Do the beneficial bacteria and fungi products that are now being marketed jumpstart that community and its activity and its functionality in a way that's economically justified?
And again, you know, it's difficult for us to know for sure. There are some cases in which adding some organisms can be beneficial. I mean, obviously, when you're working with legume plants, you need to add the appropriate inoculum. So making sure, and that's not a thing that I tell people not to do. But you also need to be looking at, like I said, sort of looking at the system and how that's going to function. But if you're looking at a soil that has been really, really depleted, or soils that have been fumigated, you could add a little bit of a biological community as sort of a short-term jumpstart. But you have to recognize that the jumpstart isn't going to replace what you could do in applying the principles to soil health.
Yeah, okay. You talked about, you know, roots in the ground all year round, green plants, all that. In a row crop, corn, soybean row crop rotation, whatever that is, during the summer growing season, can those row crops provide enough feed, food stock, carbons, and other things to optimize the microbial activity? Do you feel they can provide enough? Just corn and soybeans in rotation, just a single crop monoculture?
Unfortunately, not really, because the plants are putting carbon below ground primarily when they're in their vegetative growth phase. And so we're talking about the number of days per year, you know, and I talked about the 105 days of vacation. If you're growing crop for 260 days, now we're not growing corn or soybeans for 260 days. So you're already reducing that number. But then if you also think about the fact that they're putting carbon below ground only when they're in their vegetative growth phase, that reduces those number of days as well. And that's why we've had sort of this gradual, slow decline in microbial activity in our soils is that we haven't been able to effectively, within the rotation systems that we currently have, be able to do that. So putting in a cover crop or a companion crop can be extremely beneficial to improving the efficiency of the system.
So a corn plant, of course, at tassel, after tassel R1, what amount of stuff being leaked out of the roots goes down considerably.
Yes, yes.
Okay.
And it makes sense because the plant now is allocating more carbon into reproduction and producing the grain. So again, everything costs. You have to look at who's going to pay and how big of a price they're going to pay and when you need to pay what at what time. And reproduction costs.
Okay. Another question, this is a question I have. Is there a practical way to quantitate or to qualitate or quantitate a native mycorrhiza in our soil? Is there a practical way to do this?
You can get an idea, and I've been working on putting together sort of a compendium of different types of soil tests that you can do, including the thing with aggregate stability, but also with the mycorrhizal fungi. You can get an idea by looking at the amount of mycorrhizal fungi that are in the roots, the percentage colonization. So essentially looking inside the roots of the plant. And there is a methodology that you can utilize to be able to do that. And as I get this compendium together, I will share this information with you and all of the attendees so that they're able to get that. But essentially, you can utilize some household chemicals similar to what we use in the lab and get an idea of what might be happening in looking at colonization. Just utilizing things like drain cleaner and vinegar and ink, and be able to look at the roots. So there are ways in which you can, again, it's not exact, and that linkage between colonization and mycorrhizal activity has not ever been fully established, but at least will give you an indication of how much mycorrhizal activity you have in your soil.
Okay, I think we're kind of out of time now, Kris. Again, Kris, we want to thank you for your great presentation and talking about soil regeneration from the kind of microbial point of view. You really laid out a good argument about all these things we should be doing and how the system, we can work with the system to improve or regenerate our soils. That concludes our webinar on soil biology builds resilience, enhances soil quality. You will find this webinar and other soybean agronomic resources on the Chekhov-funded website, the Old Soil Advisor. And thank you for attending this webinar. And thank you, Kris, for the great presentation. This webinar will be posted online shortly in a few days. And have a great day and a great week. And this webinar is now over.
Thank you.